it used to be seen more in the older population, but now also we're seeing a more increase in the younger population of women. Um, it's important to study the endometrial cancer because of how many people it impacts. And um, concerning women, there's the symptoms of the disease. It's difficult in the daily life to manage the symptoms. So it's important to keep our research open and to develop new ideas to help the treatment for women. In our research, we specifically focused with the genes PTEN and ERBB2. PTEN has been seen in many human cancers outside of endometrial cancer as frequently mutated, meaning that the PTEN gene is seen as highly altered, which can contribute to the development and tumor genesis of the cancer. So we used a mouse model in our lab to research the correlation between PTEN and ERBB2. Specifically, PTEN acts as a tumor suppressor by regulating the cell growth, and ERBB2 also is responsible for regulating cell growth, and different members of the ERBB family play critical roles in the development and progression of cancer. So we used these two genes and studied them in our mouse model, specifically because their relationship is not well known, or how the two genes interact is not well studied in endometrial cancer. We found in our research that um, the ERBB2 functions as a tumor suppressor in the mice that have a PTEN mutation. So specifically, our mice, we had two study groups, double and single knockout mice. So our single knockout mice just have the PTEN mutation. We saw a much more rapid acceleration of the development of cancer compared to our double knockout mice. And they had a more slower progression, suggesting that ERBB2 is slowing the growth of endometrial cancer. Our research did have a limitation. Within our groups, we studied at different time points in my paper, and we did have a subgroup to our main group. We were unclear as to the cause about why we saw a subgroup in our one time point of mice. It was in the 2-week time point. We observed a difference in the histology between our group. One group displayed endometrial hyperplasia, which closely related to our PTEN group, whereas our other mice did not display endometrial hyperplasia. So we need to do more study and a more comprehensive molecular analysis to determine why we had a subset group. I would definitely say in the clinical setting, because so many women suffer from endometrial cancer in today, it's beneficial to be able to have more treatment options available. I hope that my research can contribute to the understanding of the molecular mechanisms of endometrial cancer, and also spur other people that have similar research interests to continue or have a new study method to be able to further identify the interactions of this disease.